Okay, I sent everyone a PowerPoint presentation, like I mentioned earlier, lymphocyte versus monocyte. Lymphocyte versus monocyte. So go through the slides, and it'll give you the answers, and hopefully you can figure out. Well, I'm sure you will after this monocyte lecture, because it's definitely different than the lymphocyte. But there are some things that you can fall into, some traps you can fall into by accidentally calling a monocyte versus a lymphocyte. Okay, let's go ahead and start this lecture. It's a much easier lecture than the lymphocyte lecture. If you have any questions on the lymphocyte lecture, just let me know and I'll clarify it for you. Let's go ahead and get started with the monocytes and macrophage lecture. Pretty simple lecture, like I said. Okay, monocytes, mononuclear phagocyte system, the MPS system. It's composed of monocytes, macrophages, and its precursors, which are the monoblasts and the promonocytes. There are not too many developmental stages for the monocytes. It's basically monoblast, promonocyte, monocyte, and macrophage, and that's basically it. That's the whole maturation sequence for monocytes. It's pretty simple. The monoblast, the youngest stage, is large eccentric nucleus. Just like most blasts, it has prominent nucleoli. NC ratio, nuclear cytoplasm ratio, is 7:1 to 4.1. Monoblasts, just like you can think about it, monoblasts are non-motile, non-phagocytic, minimal indentation, and prominent nucleoli, as with any other blast in any other stage. Myeloblast has nucleoli. Lymphoblast also has nucleoli. But if you can compare this stage, the monoblast, to like a newborn baby, they don't know how to walk yet, so it's non-motile, and they can't feed on their own, so it's non-phagocytic. So monoblasts are non-motile and non-phagocytic, but it has nucleoli. The second stage, the middle stage, are the promonocytes. Here, we're starting to see indentation or folding of the nuclei. And when I talked about the cells of the lymphocytes series, I tried to impress upon you that there's no folding in the nucleus in a lymphocyte, no folding of the nucleus in the lymphocyte. However, in the monocytes, there is nuclear folding. The chromatin is still fine in the promonocyte stage. Fine chromatin, a smooth texture of chromatin, is characteristic of an immature cell. Okay, remember that. I talked about that in the Myelocytic series. The chromatin is fine, but for promonocytes, we're starting to see indentation. Here's the monoblast. Okay, so if you look at this cytoplasm, the cytoplasm, if I was to look at this and I look at the cytoplasm, I would say it's monocytic in origin. I would not call this lymphocytic. And you'd say to me, you're crazy. I would call this a lymphocyte or lymphoblast. Okay, blast because it has nucleoli, but I don't see too much blue here in the cytoplasm. That's one thing that I look for if I'm going to call it a lymphoblast versus a monoblast. The cytoplasm is dull gray. That's one of the characteristics. Make sure you make note of that. I'm going to mention it later. Cytoplasm has a dull gray color appearance. Dull gray. All right. And not supposed to see any kind of folding or indentations, but this is a monoblast. Blast because nucleoli, also two blast because it's large. Monoblast I would question because there's usually folding here, but usually when you see indentations or folding, it's because of the cells around it. That's a monoblast. Monocytes. Monocytes is the mature stage. You got monoblast, promonocyte, and then right away, monocyte is the mature stage. Visible nuclei, this is still promonocyte. Visible nucleoli, still in the second stage of the promonocyte. Granules are positive for peroxidase, okay? Usually what in histology, they will try to figure out what cell line you're looking at. Like for example, you're not really supposed to tell the difference between an erythroblast versus a lymphoblast versus a myeloblast versus a monoblast. And the way you're supposed to do it is by special stains. But in the promonocytes, the granules will stain positive for peroxidase. Okay. Promonocytes, the NC ratio is 4:1 to 2.1, slightly motile as opposed to the monoblast. And so it's starting to move and it's starting to eat on its own. So slightly motile and it's starting phagocytosis. Promonocyte. So this is large. Promonocyte, it's pretty, you would say that this is pretty kind of bluish, but I would still see kind of gray. This is kind of gray. Once you see the grayish of a monocyte, you need to memorize it. That gray in this one is a monocytic cytoplasm. And because of the folding here, and because of the, I guess you can kind of see folding here, but definitely this is a promonocyte. Folding here, and the dull gray cytoplasm, that puts it in the monocytic series, but it's immature, and maybe there's nuclei here and here, making it immature, so it's a promonocyte. Okay. Finally, the monocyte, which is the mature stage, larger than a mature neutrophil. These are bigger than a regular neutrophil. And the monocytes are only in the circulation for a short period of time. And we already went through the developmental stage. We went from monoblast to promonocyte to monocyte. And that in that short period of time, because in the peripheral blood, normal value for monocyte 0 to 10. So you won't see that many monocytes in the peripheral blood, because after it matures from monocyte, it goes into macrophage. Okay, they migrate into the tissue at the monocytic stage and become macrophages. Remember, macrophages, what macrophages do, they're the garbage trucks of your blood. They go around and collecting garbage and debris and stroma and things like that, dead cells. Macrophages are the garbage trucks of your blood. Monocytes have abundant cytoplasm in relation to the nucleus. NC ratio is 1:1 to 2.1. A lot of cytoplasm, okay? Monocyte cytoplasm. Know these characteristics. Okay, so the next couple slides, I'm gonna give you characteristics of the monocyte cytoplasm. Make sure you know those, and they will be asked on a quiz or an exam. Dull gray blue cytoplasm, dull gray blue cytoplasm, and fine reddish or purplish stained granules. So when I was in my CLS training, they said we were described lilac granules. I said, what's lilac? I don't know what lilac means. I guess that's some kind of Avon color. Well, I didn't know. And it took me a while to figure out what a monocyte was. But when I see the descriptive term of fine reddish or purplish stained granules, you'll know what I mean. Okay, it's either reddish or purple. You actually may see both if you zoom in really close. The granules are evenly distributed, and the granules of the cytoplasm are ground glass in appearance. Ground glass in appearance. Okay, it's kind of like fine, but you see the fine reddish and purplish granules, but it's ground glass in appearance. And also too, one quick way to figure out if you have a monocyte is if you see vacuoles. If you see vacuoles, I know you can see vacuoles in neutrophils, but you know what a neutrophil looks like from last Wednesday. But monocytes are larger than neutrophils. Remember that. The vacuoles have a digestive function. So if you see vacuoles, that means it just finished lunch. In disease states, these vacuoles can contain red blood cells, contain nuclei, cell fragments, bacteria, fungus like yeast, and pigment may be present. Okay. The nucleus of a monocyte can be folded. Remember I said, as opposed to a lymphocyte, it can be kidney bean or horseshoe shape. Kidney bean or horseshoe shape. And the nuclei may also look like convolutions, like a brain. Okay, so the nucleus of a monocyte is very flexible, obviously from these descriptions, kidney bean or horseshoe shape, or convolutions like a brain, as opposed to lymphocyte. Lymphocyte nucleus is pretty rigid, okay, and it's dark. The nucleus of a monocyte is spongy looking. I didn't get into that description yet, but it could have vacuoles, okay? Here, the monocyte nucleus can be lacy, chromatin network, or kind of spongy looking. It can be lacy, okay? The nucleus as opposed to a lymphocyte. Remember, the lymphocyte is dark, coarse, and it's almost solid. It's coarse clumped. The chromatin of a nucleus is lacy. It's kind of spongy looking, okay? That's a monocyte. And it also has folds. It's not as rigid as a lymphocyte nucleus, because those are the two distinctions. Lymphocyte nucleus is hard, solid. A monocyte nucleus is flexible and lacy looking. Okay, monocytes are motile and can have blunt pseudopods, just like the reactive lymph. Remember the reactive lymph? I don't want to use that as a comparison. A reactive lymph, remember, has blue tinges. That's how you know it's a lymphocyte. Monocytes will not have those blue tinges. Okay, know these 4 descriptive features of monocytes. Nuclear convolutions, you'll be asked this. Okay, so 3 asterisks. He's gonna ask this on a quiz or an exam. Nuclear convolutions, lacy chromatin, dull gray-blue cytoplasm, and blunt pseudopods. Make sure you know those. Okay, this is a monocyte. See how it's horseshoe shaped? I actually gave this on a quiz. I didn't see the MO there. So that obviously a monocyte. So dull gray-blue cytoplasm. Okay, dull gray-blue cytoplasm. It's not a band because the cytoplasm is not do not have the secondary granules. You don't see the secondary granules, so it's not a band. Okay, so don't confuse that. Monocyte again. Okay, again, kidney bean shape, dull gray-blue cytoplasm. It's kidney bean shape. Okay, it's not a band because cytoplasm does not have the secondary granules. If it did, then it possibly could be a band, but it's not. They're not there. So monocytes are often mistaken for large lymphocytes. Monocytic cytoplasm may be blue, but generally it's not. Usually lymphocytes have kind of a blue cytoplasm. Granules in a monocyte are reddish or purplish, okay? Nucleus can be round, okay? The rules, monocytes may not follow the rules, but because I said they're kidney bean or horseshoe shaped, well, you can get a monocyte that has a round nucleus, just happen to be round, but go by the cytoplasm and make sure you see that it's dull gray in color. Blunt pseudopods and digestive granules may be missing. So this page is to illustrate that monocytes necessarily will not follow the general rules of the monocyte definition. So if you're not sure, look at the chromatin pattern, ground glass versus dark clumped, or spongy looking or lacy looking versus dark clumped. If it's dark and clumped, it's a lymphocyte. If it's ground glass, lacy, or flexible, then it's a monocyte. Look at the cytoplasm. Is it vacuolated versus clear with some bluish coloration? If it's vacuolated, then it's a monocyte. If it's clear with some bluish coloration, then it's a lymphocyte. Look at the shape of the cells or the nucleus. If it's horseshoe, it shows flexibility versus round and rigid. Horseshoe, kidney bean, showing flexibility, that's a monocyte. If it's roundish or rigid and dark, it's a lymphocyte, okay? Large lymphocyte, nuclear chromatin tends to be clumped. The chromatin is not lacy, okay? For a large lymphocyte, the chromatin is not lacy. It's a large lymphocyte. They may have large granules in the cytoplasm. Okay, a large lymphocyte, it's not illegal, and I've seen this, that large atypical lymphocytes, if I looked at the cytoplasm, oh, there's granules in there. It's not illegal for lymphocytes to have granules. Remember which lymphocyte contains granules? It's the cytotoxic T lymphocytes, okay? Large granules in the cytoplasm, but it's generally, as a rule of thumb, the cytoplasm is clear of granules. Is this a large lymphocyte? Could be atypical, but it's a large lymphocyte. Again, dark, coarse clumped. It's not spongy or lacy looking. A lot of cytoplasm there. It could be classified. You can call it, I wouldn't mark you wrong if you call this atypical. Monocyte, brain-like convolutions of the chromatin pattern, may have distinct bluish red granules. I talked about that. And fine granules in the cytoplasm give a ground glass appearance. So that's a description of a monocyte. Monocyte, obviously, see the grayish, the dull gray color, but even more so, look at the vacuoles there, and look at the convolution, how the nucleus is convoluted. Okay. Now I can see how somebody could confuse this with a neutrophil that has the filaments hidden, and it's a vacuolated neutrophil. Okay, but based on the color, I don't see the granules in the cytoplasm of a neutrophil. So, and then also too, the color, the dull gray color of the monocyte, this makes it a monocyte. Okay, once you see this, call this as a monocyte, and you see this color, memorize it, embed it in your brain. This is a monocyte nucleus. Okay. Okay, so as monocytes enter the circulation for a short period of time, they migrate into the tissue, become tissue macrophages. And I described for you what macrophages do. They don't normally re-enter into the bloodstream. So once it goes into the tissue, they don't usually come back except during intense inflammation. Okay, I've never seen a macrophage on a peripheral smear. Usually they're monocytes. If I saw a macrophage, and macrophages are huge, I'll show you that in a minute. Macrophages are huge. Okay. Where are the macrophages located? They're located in the lung, the peritoneal, the spleen, liver, and connective tissue. Okay, so what they're doing is they're just lying around until the need arises. So they're just lying around in the lung, spleen, liver, and connective tissue. Macrophages, these are really, really large cells, and you won't see them unless you're doing a body fluid smear. The nucleus may be round or kidney shaped. Generally they're round and may contain nucleoli. Abundant cytoplasm, a ton of cytoplasm with vacuoles. Well, you'll see it shortly. Numerous aerophilic granules. Once you see these cells, you'll say, those are macrophages / histiocytes. Macrophages and histiocyte are synonymous terms, just like what I just said. And macrophages are usually seen in body fluid cell counts. So this huge, humongous cell, this is a body fluid prep. It's probably a cytospin. Look at how spongy the cytoplasm. These are all, let me see. Yeah, they're all monocytes. This is a large macrophage. Okay, see how spongy the cytoplasm is? You see something that big and round, that's a macrophage / histiocyte. Same thing here. This one probably just had lunch, and that's probably either yeast or bacteria. Okay, macrophage, histiocyte. That's why it's slashed. They're synonymous terms. If you see macrophage, it's also called a histiocyte. And that's it for monocytes and macrophages. Do any of you have any questions? Make sure you know the difference between lymphocyte and monocyte. Let me see if I'll go over a few of these cells. And I sent this to you. I sent this to you on your email. I emailed it to you during your break. All right. So basically, you just hit the down button. So this would be a monocyte. This would be a monocyte for me based on the color of the cytoplasm. So that's a monocyte based on the color of the cytoplasm. I'm not going to go through the whole 73 slides, but I'm just going to go through a few of them so that you'll get a feel for how to distinguish between a monocyte versus a lymph. This is That's a lymphocyte, but I'm looking at the rouleau. See, did you guys get talked about? I don't know if you looked at the RBCs, but see all that rouleau there? That means you're too far deep in. Okay, lymphocyte. See this lymphocyte? No, yeah, that's a lymphocyte. I thought maybe I saw vacuole. These are lymphocytes. Okay, round nucleus, almost close to the size of an RBC. The color of the cytoplasm makes it a monocyte. The vacuoles makes this a monocyte. The lymphocyte rigid nucleus makes this a lymphocyte. Again, the hard roundish nucleus with, I can see how you can say, well, that has the dull gray cytoplasm of a monocyte, but look at the nucleus. It's rigid, round, coarse, dark, and clumped. So that's a lymphocyte. This is a monocyte based on the color of the cytoplasm and the vacuoles. So it's a monocyte. This is a monocyte, even though it has a bluish here, it can be misleading, but you'll never see a lymphocyte with that flexible of a nucleus. So this is a monocyte, okay? See, you can see how this can be misleading as a lymphocyte because of the blue cytoplasm here. And this is blue because it's touching the red blood cell. This is blue because it's touching these two cells here. So that's what makes it blue. But this is a monocyte based on how flexible the nucleus has become. This is a lymphocyte, pretty classic, roundish, hard, dark nucleus, lymphocyte. This is a lymphocyte also. Solid nucleus, bluish cytoplasm. These are lymphocytes, probably reactive. Reactive, remember, reactive / atypical, okay? Lymphocyte again. This could be reactive versus lymphocyte. Again, if you see how this is touching the red blood cell, there's that bluish color. Monocyte, because of the shape of the nucleus, you'll never see a lymphocyte with a horseshoe shape like that, and a lymphocyte with those kind of vacuoles. Lymphocyte again, even though. Now, one thing about lymphocytes, they can have, in the mature stage of a lymphocyte, it can have nucleoli present, but this is a lymphocyte, solid chromatin and clear cytoplasm. This is a monocyte based on the folding here and the vacuoles. So this is a monocyte. Okay, so you guys get the idea. Go through these, and maybe I'll give you an exercise on this down the road. Okay, so this is for you. Who was it that asked lymphocyte versus monocyte? Was that you, Dan, or Jeremy?
It was me, Dan.
Okay, so now with this exercise, does that help clarify lymph versus monocyte?
It does. Thank you.
Okay, so go through these, and I'll give you perhaps. I mean, it's hard. Even at the CLS level, I had trouble monocyte versus lymphocyte, and I didn't have an exercise like this that went through the different cell types. But I went through a lot of descriptive characteristics of both lymphocyte versus monocyte. Just remember, lymphocyte is a solid nucleus, dark, and not flexible, versus a monocyte. You got flexibility and you have vacuoles. Okay. All right. That is the monocyte lecture. So next, I already went through exercise 10. Hopefully that the explanation I gave you was pretty straightforward. Now I'm going to all right. I am going to stop my video, but I'm going to keep my stay on audio. And I'm going to review the quiz that you'll be getting tomorrow, okay? So you got case studies, and all of the case studies that you had in homework 10 was on bad samples, how to identify bad samples. Now you know, and please look at my explanation of CBCs, of the 5 CBCs. One was normal, one was clotted, one was cold agglutinins, one was platelet clumping, and the other one was lipemic icteric. So make sure you know that what you're looking at is reportable or not. And the first thing you look at is look at the MCHC. If it's not, then that means it's a bad sample. And then you need to tell me why it's a bad sample. And one of the easiest things to do is look at the 3 cell lines. What's going on with the whites, what's going on with the reds, and what's going on with the platelets. If everything is out, and okay, so if all of them are low, and if you had an H&H of 12 and 37, is that anemia? Is that the aplastic anemia and everything's low? No, it's not. It's not anemia. So that's a clotted sample. And also to the MCV. Look at your MCV. What are you feeding through the flow cell? Okay. So for quiz 10 tomorrow, you're going to be getting case studies. And it could be a combination between a bad sample versus a hematological problem. So you will, okay, so you guys know what causes eosinophils to go high, right? What causes eosinophils to be elevated?
Parasitic infection.
Right. And when you have a parasitic infection, what kind of anemia is that? We talked about anemia, so I can tell you that it's an anemia. What kind of anemia? Pardon me?
Pernicious.
Yes, pernicious. Due to what deficiency?
B12.
Right. Okay, make sure you say that.
Got it.
All right. I'm going to give you a CBC that fails, and based on my explanation of the 5 CBCs at the beginning of the lecture, you're going to need to be able to tell me what the problem is, okay? I think I was pretty direct in my explanation. So make sure you go over why it's clotted, why it's cold agglutinins, why it's platelet clumping, and why it's lipemic icteric. And how to correct. You know how to correct. Like platelet clumping is redraw sodium citrate, lipemic icteric is saline replacement, clotted is redraw the patient, and cold agglutinins, where only the reds are down, is pre-warming. What would you suspect if you had a total protein? Do you guys know the normal value for total protein? I think it's like 6 to 8 grams per deciliter. But if you had a total protein of 12 or 13, what would that tell you?
Is it proteinuria?
Proteinuria? Okay,
Yeah.
what about in the blood? I'm not interested in the urine.
Hmm.
What disease are we talking about?
Inflammation?
More specific. Elevated protein.
Congestive heart failure?
No. Begins with a W.
White blood cells?
White blood cells, not a disease.
Strong.
You better not identify yourself. W, white blood cell. Okay. Waldenstrom's. Waldenstrom's. Okay, what's Waldenstrom's? Elevated what? Elevated protein. Multiple myeloma, elevated protein. Make sure you know what causes elevated protein. That's the one where I mentioned during lecture that you spin it down and it's still pretty clear, but it's protein that's causing the MCHC to be high. So you do a saline replacement on that elevated protein plasma, and then everything will come down. It's because of the elevated protein. Like if the patient has Waldenstrom's or multiple myeloma, you need to,
Why the-
that will cause the MCHC to be elevated, and it won't be between 32 and 36. You'll get something in the 40s or high 30s, okay? Somebody had a question? I can't hear you.
Sorry, that's my background noise.
Okay. All right. Make sure that you look at the rule of threes. Make sure you look at the MCHC. Make sure if it's reportable, then you have a disease. You're dealing with a disease, some kind of anemia. If it's reportable, if it's not, then you're dealing with an artifact. That's how you'll know. Okay. That's how you'll know the MCHC. If the MCHC is out of whack, then you're dealing with an artifact. If it's in there, then you have some type of anemia going on. Okay. Do you guys know how to tell the difference between thalassemia and iron deficiency anemia? What type of anemia are both of them?
Hypomicro.
Correct. Both of them are hypomicro. But what's the difference between, give me two differences between thalassemia and iron deficiency anemia. Thalassemia will have what?
The bell-shaped,
Say again?
bell-shaped cells.
What kind of cells?
The bell, like the target cells. Are they the target cell?
Yes, target cell. The bell is a wet mount. We're not going to do wet mount preps. That's a natural shape. But target cells, okay?
Okay.
All right. So thalassemia will have the target cells. Anemia will have, iron deficiency anemia will have everything, okay? But another way to tell the difference between thalassemia and iron deficiency anemia, other than the target cells in thalassemia, is by looking at the RDW. Okay, both of them have an ESO. Both of them have an ESO, but iron deficiency anemia has a much higher ESO, probably in the 20s. Okay, iron deficiency anemia has RDW in the 20s, so it has a lot of an ESO and a lot of POIC. Thalassemia is going to have POIC, the target cells, but the amount of cytosis in thalassemia is not as high as iron deficiency anemia. It'll probably be in mid-teens, like 15, 16, 17. Okay, so that's how you tell the difference between thalassemia and iron deficiency anemia is the POIC. Thalassemia is mainly targets, and the RDW. Iron deficiency anemia has a much higher ANISO or RDW. Okay, that's how you tell the difference between the two. Where do you see oval macrocytes and hypersegmented neutrophils? Anybody?
Is
B12
it
deficiency.
myeloplastic? Oh, just kidding.
Say that again.
B12 deficiency.
Yeah, B12 deficiency. Okay, so for aplastic anemia, everything is low, correct?
Yes.
Yes. And because it's anemia, what can you tell me about the retic count?
It's high.
Oh, really?
It's less than 1%
Why is that?
because of the hypocellular.
It's hypo, yeah. Hypoproliferative bone marrow. So remember, aplastic anemia, the factory is shut down. Everybody, whites, reds, and platelets. So if you're not making reds, you're not making retics. So if you're not making retics, then your retic count is going to be low. Okay, so you have an anemia, but your body doesn't care. So because there's nothing going on in your bone marrow. Okay, make sure you know that. In aplastic anemia, just because you have anemia, normal response to anemia is to make more red blood cells, but not in aplastic anemia. That's the defect. You're going to get, if you have aplastic anemia and you don't treat it, you're going to die. Okay, because you need blood to survive. So, but in aplastic anemia, the factory is shut down. Your whites, reds, and platelets are not working. So you're not making retics. So therefore your retic count is low. Did I ever tell you what the difference? I know I'm going to do it next semester when I talk about malaria. But how you can tell how you can identify plasmodium? You know what plasmodium is, right? That's the malaria organism. But how do you know? And it's important to know that you're dealing with Plasmodium falciparum because of the 4 species. Plasmodium falciparum is the killer. Okay, you want to be able to identify Plasmodium falciparum. Did I ever say what identifies Plasmodium falciparum?
I don't remember.
Okay, so you know malaria, right? Malaria, you'll see the ring forms, right? You can identify malaria by identifying the ring forms. For Plasmodium falciparum, within one red blood cell, you can see multiple ring forms. You can see more than one ring form in a red blood cell, and that's how you can tell Plasmodium falciparum. So the term multiple ring forms is definitive. All right. That's it for quiz 10. Quiz 11 is the Myelocytic series. Make sure you know that the characteristics of the different stages of the cells in the Myelocytic series, which one has a high, starting from youngest to mature. Remember, you start out large and blue and not complex, and then you increase in complexity and your nucleus becomes more coarse and clumped. Our rods can be in the first 2 stages, the blast and the promyelocyte. And know the difference between primary specific and secondary specific granules. Know how to distinguish between a band and a metamyelocyte. In the amount of indentation, if the indentation is deep past the midpoint, it's a band. If it's not quite towards the midpoint, then it's a metamyelocyte. Know about the Golgi apparatus. Know where what stage the Golgi apparatus is identified in. How long does it take to get from blast to mature neutrophil? I mentioned that in the lecture. Talked about left shift and right shift. Left shift means you're going left and you'll see more bands. Right shift, you're going right and you'll see hypersegmented neutrophils. So if you have a left shift, you'll see more bands, but you'll also see more neutrophils as well. Okay, left shift, your body's responding to an infection, so you're going to have neutrophilia, but you're also going to see immature neutrophils or bands. So you'll see both bands and neutrophils. Okay, our Pelger-Hewitt cells, is the nucleus mature or immature?
They are mature.
Say that again, one more time.
They are mature still.
They're mature. They are
Yeah.
mature. They're not immature. They are mature cells that only have 2 lobes. Okay, bands have 2 lobes, but they're immature. That's how you can tell the difference.
What's that called again?
Pelger-Hewitt or hyposegmentation. It's in the hyposegmentation section of the lecture. So the nucleus is mature, but it'll only create 2 lobes. That's why you have the sunglasses. But they're mature. The chromatin is mature, as opposed to a band. Bands can be bilobed, but it's not as mature. The chromatin is not as mature as a mature neutrophil. All right. Know what causes eosinophilia besides parasitic infection? And know the composition of the RBC inclusions, like Dolly bodies. What are Dolly bodies made of? And know what's seen in inflammation or infection. You'll see the 3 things. Remember, toxic granulation, Dolly bodies, and vacuolated neutrophils. All right. And that's it for quiz 11. It's pretty simple. Just make sure you go over the lecture, the PowerPoint lecture, a few times, not just once on the day of the quiz. Please put some time into studying. Okay, those are the 2 quizzes. And now I'm going to go over on the exam portion. I went over the lecture portion last week. Okay, so if you want a review of the lecture portion, listen to the recording. But I didn't go over the matching portion. So on the matching portion of exam 2 next week, make sure you know the different shapes of the RBCs. Like somebody mentioned bowl shape. Something's bowl shape, something is biconcave disc, something is bell-shaped curve. Make sure you know those, okay? In different shapes. Of the insects and the parasites, make sure you know what vector they come from. Like, for example, malaria is obviously mosquito. B12 deficiency, the organism that causes that is D. latum, for example. Make sure you know the tests. Make sure you know what the osmotic fragility test is for. The reagents like Drapkin's reagent, sodium metabisulfite, acetylphenhydrazine. Make sure you know those reagents and what they're testing for. Okay. Make sure you know what the Donath-Landsteiner antibody, what autoimmune hemolytic anemia that's found in. Sodium metabisulfite. Anti-D versus anti-I. I talked about those 2 antibodies. Anti-D is seen in a certain disease, and anti-I is seen in a certain disease. Okay. And then the categories of improperly submitted samples, like the clotted sample, the cold agglutinin, the lipemic icteric. What do you actually see? Okay, or what are the corrective actions for each of those? Again, the composition of the inclusion bodies. So what's Pappenheimer bodies, Cabot's ring, Hall-Jolly bodies, and Heinz bodies, etc. And then the different types of anemia. Make sure you know, like Fanconi's, G6PD. What do you associate G6PD? What are the 3 things? Like G6PD, you see bite cells. G6PD, you see Heinz bodies, for example. Intrinsic factor. What do you know about intrinsic factor? Where does that come from? It comes from the stomach, but where specifically in the stomach? You know, and that's parietal cells. know where you get your B12 folate from? B12 is from the meats, and folic acid is from the leafy green vegetables. And that's pretty much it for the matching. If you can get those, it's basically association of certain terms with other terms. And if you usually in previous classes, people did well on the matching. Okay, so that's your review from the quizzes for the quizzes and for exam 2 tomorrow. So I'll be sending those to Xavier tomorrow, and he'll be giving those to you in lab. And then now I'm gonna send you a quiz, and hopefully you guys have looked at those cells. It's the abnormal RBC. All you have to do is in the header type in what you see. I'm gonna email it to you. Once you're done, email it to me back with your answers. All right. Make sure you put your names on them. If there aren't any questions, I think that's it. Does anybody have any other questions?
Yeah, how many questions is it?
How many questions? All right. You want to know how many questions? All right, for the matching. I mean for the matching. Okay, for the multiple choice. Multiple choice are 60 questions. But on the last 2 case studies, which I'm going to give to you as bonus questions, there are multiple questions. There are 4 questions for each case study question, but 60 questions in total for the exam. For the matching, so that's 60, and then matching is let's see. Okay, so for the matching, there are 2, 3, 4, 5, 6, 7, 2, 4, 8, 10. 34 matching terms. Okay, that's exam 2. For the quizzes, they're pretty short, about 10 to 15. So the quiz shouldn't be too bad. So now you have an idea for exam 2, multiple choice and matching with a couple case studies. Anything else? Okay, so I'm going to be sending you this abnormal RBC quiz. All you have to do is identify the cell. Once you finish, then you can just email it back to me. All right. All right, good luck on your quizzes and exams tomorrow, and I'll see you next Wednesday.
Have a good night.
You too, thanks.
Bye.
Bye.
Professor,
Yes.
um,